We're going to open the meeting. You ready, Tara? Okay. Welcome to the Sussex County Creation Science Club. This is our first meeting of the month of November. Our next meeting is going to be December, the first Friday in December.
December 2nd.
December 2nd. Thank you. Hey, Roseanne.
Hey.
Nice job Thursday night.
What's that?
Nice job at the theater.
Oh, you were there?
I read about it.
Oh. I'll be glad when this is over.
Hannah, would you do me a favor, honey? Would you open us up in prayer? Real loud so we all can hear.
She's a little uncomfortable doing that in front of all of you.
Oh, okay.
She'll pass.
I need help. Who can help me open up in prayer? I'm feeling something back there, like somebody wants to do it.
I will.
Okay, thank you.
Dear Lord, we thank you for this opportunity to come together tonight to learn about your creation. I am just in awe of all the ways that you show your love for us in nature and in creation. And I thank you for the opportunity to come to this club and ask for your blessing on the speaker tonight. And I pray that you will open our hearts and minds to all that you have for us tonight. Pray in Jesus' name. Amen.
Thank you. Well, welcome everyone. This is the Sussex County Creation Science Club. We meet on the first Friday night of each month, and today no different. Today is November 4th. Is it 4th? We're meeting. Come on in, guys. Come on. Lots of seats. Our next meeting date is December 2nd, the first Friday night in December. But there's going to be a change in the rooms. It seems that the ladies at the church are going to use this room, so they asked me if we would move, so I said okay, we'd move directly downstairs to one of the larger rooms. So just make a mental note that we'll be downstairs on the 2nd. Now, I'm going to try to get a speaker to come in, as we have been doing, but if I can't in a short notice, only because we were out, Terry and I had no electric for the whole entire week. We got it back at 4 o'clock. I would have been working on that. So it's been a tough week, to say the least. Well, anyway, getting back to the club here, I'm going to either show a film or get a speaker. Now, the speaker I have in mind will be on, if he accepts, his name is Dr. Cuazzo, and I haven't asked him yet, and he would probably be speaking on the origins of man. And Roseanne knows him very well. There's been some information out on another Australopithecine, which doesn't really bode well for the evolutionists, but it's 2 years old, but they're now making this Australopithecine something new. They're trying to make it new and look like, here we found another missing link, when Australopithecines are apes or monkeys. They have all the attributes of apes and monkeys. But if you want them to be a link, they could be a link if you want it to be. But that's only if you want to. But if you look at the facts, they don't have it. I thought tonight somebody might ask me about that, and that's why I brought it up, because it just came out in September. Okay, if not, we'll go on. I want to introduce this evening Dr. Tony Johns. Now, Doc here, I've known him at church. He has spoken with us in Indiana with Roseanne and I at the Indiana Creation Expo, and he did such a fantastic job that all we got was compliments from him. He knows mostly in the area of genetics, which he'll be speaking with you tonight about tonight. Now let me read his bio. I think you're going to be impressed with this. See, as creationists, we're always accused of having like this foo-foo science background. You know, we come from a religious background. Well, yeah, we do believe in God, and we believe in God, our Lord made us in 6 days. That's true. But we also know that there's science, because we know that God made the science. And we know the science that reflects a a supernatural creation, and it destroys evolution. So listen to this bio from a Dr. Tony Johns. BSc in Physiological Biochemistry, Joint Honors, Queen Elizabeth College, University of London. A PhD, Pharmacology, Welsh National School of Medicine, Cardiff, Wales. I think you can tell that by his accent tonight. Postdoctoral fellowships in Reproductive and Cardiovascular Pharmacology, University of Alberta, Canada. Postdoctoral fellowship in Molecular Pharmacology, University of Miami. Assistant Professor of Obstetrics and Gynecology, University of Texas. Assistant Professor, Physiology, University of Saskatchewan. Assistant Professor of Molecular Pharmacology, University of Miami. Section Head of Vascular Biology, Berkeley Bioscience, Richmond, California. Director of Global Medicine Affairs, Cardio Aspirin, Morristown, New Jersey. Global Clinical Lead Arrive Trial, 5 years, 12,000 patients, 4 books, 12 chapters in other books, over 100 papers, and over 60 abstracts, oral and posters at scientific meetings. Now that is a bio. And that is a creationist. Do we make our point here tonight? Well, Dr. Tony Johns is going to speak to you about the origins of life and how it is impossible for us to evolve, but we were supernaturally created. So with that, let's give Dr. Tony Johns the welcome hand.
Thanks.
Want me to get, I'll move this?
Yeah, move.
Okay.
Hi. Yeah, so all that stuff took a long time. I think the best part of being, apart from being in Britain where the schools are run by the state and the head of state is also the head of the church, so prayer is allowed in schools in Britain. And having an amazing biology teacher who took his time to point out how well everything was designed and pointed out to all in the classrooms that it was designed by God. And was always amazed at how complex things were when you started looking at them. And always told us that if Darwin had had a proper microscope, he would never have written the book, because he thought a cell was a small square of protoplasm and didn't know the complexity within the cell. But tonight what I wanted to do was talk, rather than going through everything with the original light, which takes a long time because it's so complex that you will see that it could not possibly have occurred by chance. But just go through one or two of the things that lead people to think that it could possibly have occurred by chance. In other words, chemical evolution, as they call it, or the production of first organic molecules, or the anti-law of the second law of thermodynamics, where actual disorder becomes order, which is against the second law of thermodynamics. And what I wanted to start with is what I consider to be one of the most arrogant statements I've ever read, which is our friend Richard Dawkins. And this, I think, shows that normal scientists and who profess to have open minds, and these scientists very early on in the centuries came from religious backgrounds, and Newton was a great believer in God, are against people like Richard Dawkins, who has a very, very closed mind and will do anything and say things that are obviously insults to people who are religious and to other scientists. And in Britain, he's not considered to be that important because no one really listens to people from Oxford and Cambridge there anyway. I mean, this is this is the area where people go to sort of more or less retire from science. And to say that anyone who believes in evolution is ignorant, stupid, or insane, or wicked, I think is so against any logic, because, as everyone knows, there is no reason for morality to exist except for to come from God, to come from our conscience, which was given to us by God. Anyway, there's a quote in the Bible which is really against Richard Dawkins, which is in Jeremiah. It says, "Call to me and I will answer you and show you great and mighty things which you do not know." And this is what happens when we all go to school to learn about science. God shows us exactly what is there. And it's people like Richard Dawkins that don't have the mind's eye or the ability to see things that are in front of them, and go to great lengths to try and come up with absurd theories that contain so many assumptions that they're not really science at all. Science is to observe things and to draw conclusions from the observations, and to make a simple assumption as to how things have occurred. And the easiest thing to understand is the Bible. So if we believe that the origin of life was not important, and it's something that Darwin basically left out of his books. He already assumed that there was life and that it evolved. So before evolution, there has to be something to evolve. There has to be an animal, in Darwin's point of view, or a cell, or a microorganism. So before Darwin's evolution, there has to be life. And we're told in the Bible that in the beginning was the Word was with God, and the Word was God. This is John 1, very, very famous saying. And according to creation that is laid out by God, vegetation came on day 3. Living creatures, the first animals, as opposed to plants, came on day 5. And on day 6 came the land creatures. So this is a very simple way of explaining where life came from. However, scientists, and I use this word in a derogatory term now, scientists that do not believe in creation of life have gone to great depths and great lengths to try and find ways of explaining how life began. And the new icons of evolution, these are the way that the closed-minded scientists tend to come up with new ways of explaining things, is to come up with these really weird names of animals that existed without any evidence that they exist at all. Luca is the last universal common ancestor. This is the number one animal that apparently Darwin thinks came into existence, and then from that everything evolved. And then again, it's also called the Ida, which is the initial Darwin ancestor. And evolutionists and evolutionary scientists believe that in the beginning was Luca and Ida, not the Word. However, there are no real theories put forward by anyone as to where Luca and Ida came from. They seem to go over this system a few times and come up with very strange ideas. I know that Dawkins and now thinks that life may have come from another planet, but that's basically kicking the can down the road. I mean, basically, if it came from another planet, then somewhere on the planet life would have had started there. Hawkins, who is a physicist and should day being a physicist rather than pretending to be a biologist, has come up with a G theory now where he thinks that he can take little pieces of any theory and stick them together without any evidence for anything. And he is obviously now starting to lose it because the latest book that he's put out with the word design in the title is nothing about a designer or designed life. What it's about is life, again, coming from another planet and being seeded on the Earth. And the only reason that people use these arguments is that there's not enough time on even the worst physicists that seem to think that the Earth is exceptionally old. Even at the exceptionally old Earth, there's still not enough time for life to have started, because it's too out of logic. The other thing that is problematic now with Dawkins is that he is now no longer using logic in his books. So he's gone away from logistics, which is a definite problem for a physicist. So what do we need? So if we step back and we think about what life is, life of course is a cell, and there's basic chemicals that we have to have in order for life to exist. And what I put here was the basic materials that are required for life. So we need amino acids, which are complex molecules, and they're building blocks of proteins. Proteins are much larger molecules that do all the work in the cell. We need nuclear bases, which are the building blocks of DNA and RNA, and also the building block of the universal energy of cells. All cells use ATP. I don't know of any exception to a living cell that can reproduce that doesn't use ATP. Plants use ATP, and though they can use photonic energy, but they all make it into things. You can think of one? Okay.
Well,
viruses don't have ATP, but they use other cells. So.
Well, I can think of some rather vain speculation. It came from someone affiliated with the National Aeronautics and Space Administration. They they thought they'd found some organism in a lake that had a very high concentration of arsenic, so that it used adenosine triarsenate.
Oh, right.
That turned out that didn't go over very well, and it didn't pan out.
Well, I guess anything with a phosphate group could be used. I know that there are some organisms that, as a food source, can use hydrogen sulfide because they live in vents in near volcanoes, and they've adapted. I won't use the word evolved, but they've adapted to be able to use that as a source of energy. But normally they use that, but then they convert it into ATP within the cell. So, but yeah, I guess phosphate. There's no real substitute for the phosphate bond. It's one of the best bonds for containing energy, and ATP uses three bonds there. But animals can use single bonds, or they can use adenosine monophosphate, diphosphate, or triphosphate. Each of the bonds contains energy. However, in order to release the energy, it takes different enzymes to get the energy out of the other forms of ATP. But I don't know whether any animal uses, for instance, GTP or TTP or any of the other nucleic bases. Now, if we assume that basic materials of life have to be in existence before life occurs, as the evolutionists think, then we have to think of where these came from, because they have to either form from the constituent parts, and amino acids contain nitrogen, and they contain carbon and oxygen and hydrogen. Nucleic bases contain all those as well. Lipids are important because lipids are the building blocks of membranes. Membranes, again, are universal for cells. Most cells have a bag around them, which is a membrane, and it's made up of lipid. But it's a very, very different lipid from the lipids that we know. It's not really just fat. It's fat that always can sit on water and has a phosphate group, so it has a water-liking side and a water-hating side. So it forms very small bubbles. And inside these bubbles is where the cell is, or the inside of the cell is. But, you know, being interested in using these in the industries that I work in the pharmacology industry, I'd have to admit it's very, very difficult to make these lipids. We used to try all sorts of things to make these lipids small enough and to look like baby cells. And there was one doctor when I was at the University of Miami who invented a liposome system, which was a way of making lipids into small cells. Anna?
Even the lipid bilayer in a typical cell, it's so complicated because it also has all these channels and pores that it's all regulated. So it's not just, it's not just a lipid, it's the whole structure.
Yeah, there are lots of proteins floating around in rafts on the lipids as well, and they move around and they have special functions. You're right. So it's not just it's just not fat.
And receptors.
Yeah. And without cholesterol as well, these lipids will not form proper bilayer. So they require cholesterol. So which is a different type of lipid. So that means that if we were to, for instance, if we wanted to make our own cell, we could go out and we could buy all the amino acids from a company and all the nucleic bases and all the lipids and the ATP. However, putting something like that together would be more or less impossible.
Besides which, God says, get your own dirt.
That's right, yeah. That's right. I saw a nasty quote on that, which was anti. So let's start with amino acids. So we only use 20 amino acids. So there are a few amino acids that other organisms use that are different to ours, but we use more or less 20 amino acids. However, there are 390 known amino acids. So if you were to look at the number of amino acids, only 20 are used. And each amino acid, if you were to form it, could be formed in two different ways. There's basically a right-handed amino acid and a left-handed amino acid. However, in all animals that I know of, and plants as well, only the left version is used. The right version we use in the pharmaceutical industry as drugs, because they cannot be used by the body. The same with sugars. With sugars we use the opposite with sugars. In sugars, only the R form of sugar is used. So if you use the L form of sugar, it can't be used by the body, so it still can be sweet and can be an artificial sweetener. And some of the artificial sweeteners use this trick of the chemists to use L form sugars. And I guess in the drug industry we use R formed amino acids to block protein synthesis when we want to. And the nucleic bases are very complicated. So ATP is basically made from adenine, which is the top left-hand one, and then there are the different nucleic bases. There are 5 nucleic bases. In DNA we use everything except uracil, and RNA we use uracil. But these are fairly complex molecules. So how would the let me just show you this again goes to show you that only the right-handed sugars, the D forms of sugar, are used and not the L forms. These are the nucleotides. So if you take the adenosine and the nucleic bases that I just showed you, and you add a sugar to them, and then one phosphate group, they become nucleotides. And these are used to make DNA and RNA. But I'm not going to get into that because that would take another lecture to talk about the genetic code. And that would go into a very circular argument because it's very difficult for both the evolutionists to understand how the genetic code could have arisen. In other words, whether the genetic code was made from proteins, from the cell, or whether the genetic code just appeared and then it made a cell, or how the genetic code could have known what proteins you needed for a cell. In other words, how it would have intelligence. So and I'll get into that a little bit later. So these again now are basics of life. So you can see as I go through this, these things are getting more and more complicated. Now we have phosphorus as well, and we have nitrogen, and we have oxygen, and carbon. And some amino acids, which I talked about earlier, also have sulfur in them. And that's also a problem for evolutionists to understand where the sulfur came from. And they're required for what they call secondary and tertiary structure, the structure of the proteins. So in the 20s was a very, very famous experiment, the Miller-Urey experiment. And this basically assumed that the basics of life could be made from inorganic things by passing an electric current and make a primordial soup. And I'm sure that all of you have heard these terms, the primordial soup, the first living thing, the Miller-Urey experiment. In fact, it's still in textbooks, this experiment. And it captured the imagination of many scientists. This was the answer to the origin of life that Darwin had never got to. Because what they did was they had an apparatus, which I think I've got on the next slide. They put an apparatus together which they said was very similar to the atmosphere on Earth when the Earth was formed. So it contained ammonia and water and nitrogen and probably carbon dioxide as well as a carbon source. They passed a large electric current in the top right-hand globe. They boiled water on the bottom to give it a nice hot soup-like texture. They put in a condenser, which is just underneath the chamber there, to make everything back into a liquid. And they made this cycle with this high voltage spark going through. And they sealed this, and they ran it for, I think it was 2 weeks they ran this. Then they analyzed what they got in the soup at the bottom.
Well, I noticed they don't have oxygen there, and I'm assuming they're claiming oxygen came later. If there was oxygen there, wouldn't that be harmful to the organisms at that stage?
Yeah, and there was oxygen basically came from the water for the amino acids. This was their argument. There was no oxygen in the system because they thought that the primordial Earth didn't contain oxygen. Whereas the amount of oxygen has actually been going down since we've been measuring it rather than up. And if you were to believe the geology samples, and if you believe that what some scientists are saying it's true, when they measured the bubbles in amber, for instance, which is a a resin, and we went out looking for amber, right?
Yes.
The air that's trapped in amber has got a much higher oxygen content than it does at
32?
the moment.
Was it 32%?
I think it was much higher. It's 21% now, so it's in the 30% range.
It was
for
in
sure.
the low 30s
Yeah.
compared to 21% today.
Yeah.
So things are not getting better.
No, and I mean the argument would be that amber seems to concentrate oxygen, which of course it doesn't, because it had been there a long time. Of course, it depends on how old you think amber is as well. But you know, even if it's older than the tree it came from, even if it was 2,000 years old, there was more oxygen around when amber was formed. So the oxygen here would not be that much of a problem in this apparatus, I don't think. It would be a problem if you ever wanted to explain where RNA and DNA, the code of life, came from, because they don't like oxygen at all. RNA especially, it gets wiped out by any oxygen. The other thing is, where would this have occurred? So where would there have been lightning storms with an ammonia atmosphere? The evolutionists tend to argue that this occurred in small caves somewhere in isolated areas, and the soup drained into a large, like, cauldron, like a piece of rock, and that this could have occurred. But this is, I don't think, very likely. I mean, it's an interesting experiment, and lo and behold, they did find the building blocks of what I was showing you. They showed some nucleic acids, even though they weren't nucleic acids that were used. Interestingly, they found a lot of formaldehyde. Formaldehyde is very easy to be made this way, which would have killed everything, because it's incompatible with life. So they tended to sort of gloss over the formaldehyde levels.
I don't see that listed.
No, it's not there. It's in most textbooks, they don't mention the formaldehyde. But that's one of the simplest. That and urea is probably the two easiest organic compounds to make.
And they're both toxic.
Well, urea isn't toxic, but it's not good. It's we get rid of urea very, very much because it's a breakdown product of proteins, so and of amino acids. So it's one of the ways of getting rid of old amino acids. So you'd imagine that the amino acids that are made will, if they keep going through sparks, if it gets going through the system over and over again, that it will be broken down to a simpler form. Follow the law of.
You say that there's proteins, but when I remember reading about it, no proteins that were formed.
No, it's only the
Strings.
building blocks. Yeah.
Yeah, there are only strings of amino acids, which in themselves don't necessarily
Yeah.
make a protein.
Well, it depends on your definition of a protein. We used to consider anything below 10 amino acids a peptide.
Right.
So above 10, would it be a protein? Maybe.
What if it
Yeah.
wasn't at that time?
It'd be a peptide if it was like below 10 amino acids. So if it's above 10, I would call it a polypeptide. So then a protein would be perhaps 100 amino acids. They didn't find true proteins.
In order to be a protein, it needs to have the alpha helices and the beta pleated sheets. These in turn must have a proper orientation, to say nothing of subunits coming together.
That's right. And not only that, in their experiment there's no sulfur. So there wouldn't have been any secondary structure, because all secondary protein structure.
Methionine,
Use methionine, cysteine.
Cystine.
Cystine, yeah.
Yeah.
So that's a pile of bricks and sheetrock and plumbing parts and no house.
That's right. Yeah, for sure. But this was, there was some glycerol was made, which of course is soap, which is again not really compatible with fats, but at least there were some fatty acids. There was no phosphorus in the experiment, so there were no phospholipids. So they would have had to add a lot of other things. They would have had to add a source of sulfur, a source of phosphorus. And I don't know whether that was ever done. You know, I don't know whether, I mean, I know that the experiment was repeated and has been repeated many times. And each time it's repeated, they find new things. You know, this is where I got some of the information on the formaldehyde and the urea. But I don't know whether it's really, somebody's really gone to town, because I think that most scientists thought it was very interesting at the time, but then forgot it. The only people that didn't forget it is the people writing textbooks.
Didn't Carl Sagan spend a number of years trying to develop this?
Yeah, I think they never got past what was first seen. I think everything that Sagan or the people that repeated experiments found was worse things that were being formed. And you know, the amino acids that were made would have been probably very simple amino acids. And I couldn't find anything that listed, you know, out of the 390, or I would be more interested in the 20 that in the human, how many of those were there. I would expect them all to be, or half of them to be D-type and half to be L-type, because there's nothing driving them to being the left-handed or the right-handed amino acids. So already we have twice as much stuff as we need. So we've got pieces of wood, but we've pieces of wood shaped one way and the other way. So, you know, curved in and curved out. If we were making a house, we would have parts that half of them would be wrong and half of them would be right.
When you say 20 amino acids, not like humans, but like are all animal type? Do we
No,
all
that-
use the same 20?
Well, most of the amino acids they use are there's about 24 that are used by living animals. Plants use about 18.
A different 18 or a subset of
They're a
the same
subset of the 24. Some organisms make for defense make some amino acids. There are some L amino acids, D amino acids as well, to defend themselves against other animals, or to make themselves not palatable, or to kill other things, or whatever they were designed to do. But I think mostly it's about 24, perhaps 25 amino acids. But I don't think every animal and plant has been checked. The same as not every animal and plant's DNA has been checked to see whether it's exactly the same. It's assumed that it's very similar among species, but it's very difficult to know whether DNA is exactly the same. The coding part of the DNA is exactly the same from one human to the other.
I don't know if you recall, Dr. Bob, I believe, made an experiment where he had a small biosphere and he tried to mimic what he felt was the early atmosphere. He did increase the oxygen content to 32%. And I thought he put a copperhead snake in there, in the biosphere, and when the time he took it out, it went from poisonous, to non-venomous.
Well, he didn't say, he assumed that it would be, he didn't get to test that. He said, but the structure was different.
The structure of the venom, where it was crooked and spaghetti-like, it was just lined up very straight.
Well, why?
So I guess the point he was probably making is, maybe the atmosphere was conducive to not having venomous animals at the time,
Yeah,
when everything was created very good.
yeah,
I mean, you gotta
First
connect,
wasn't there.
yeah,
you gotta connect, the pictures here.
yeah. I mean, it's the same. It goes to the same as the oxygen theory, where if there was more oxygen, things could be bigger. You know, which has been used to explain giants and dinosaurs and stuff like this. Is that
But
if
Bob, it wasn't just oxygen that he put into that.
What else?
He changed the magnetic field
That-
and increased it, and that was, I think that's what it.
Yeah. Well, I mean, lots of things will change how proteins are put together. So,
Didn't he experiment with healing properties, cuts,
Hydrogen? No, wait,
didn't he, a broken arm or something, didn't he?
no, I don't remember that.
Isn't this a great leap anyway? I mean, when you look at it, there's no
Yeah.
carbon dioxide, there's no ammonia in the atmosphere,
Yeah.
there's no salt. Where did those things come from?
Yeah.
This is a leap right
Yeah.
into this thing. And then you combine them, all the things we have, we know that make up amino acids and protein, and then we put them all into place, and we say, well, okay, we created life, but they never did anyway.
Yeah.
There's still
And since there was, and even if they had produced, even if a protein had been produced, the formaldehyde would have denatured it anyway. It would have had no, it would have looked like egg white. It would have been solid.
one protein is nothing, because the
Exactly.
average cell has 2,000 proteins, they're different.
Yeah. But this then goes to the fact that what they say is that if you do this experiment long enough,
Yeah.
so it's the old argument is that if you do it for a long enough period of time, you know, then eventually it's like the monkeys can write the Bible or the books of Shakespeare if you give them typewriters and you have enough monkeys. You know, the trouble is that the world doesn't contain that many monkeys to start off with, so you wouldn't be able to do the experiment. So it's a great experiment to do, to suggest, because it can never be done.
Well, it's like Telhard de Cauldron's study where apes should be, that he's claiming that there's some parts of the world they've turned into humans, but there's never been any fossils to
Yeah.
verify.
They claim anything if you don't
Yeah.
need to verify it.
Yeah.
You know,
So because they've never been able to produce life in this way, this is where we're getting it's come from outer space.
Yeah.
Yeah,
Right.
I have to go that way.
Yeah, because but they still think it came this way. It's just that they need more time. They think they have more time, and you know it's. I don't know, this just builds so to more or less say what I got. So none of the building blocks, even though they found these building blocks, and this just goes to what everyone said here, then none of them were formed into long strings or any structure whatsoever that would be believable. There was no ATP found, there was no real sugars that could be used. Everything was mixed up. There were very small amounts of everything present. I'm sure that when they did the mass spec at the end of these experiments, they found all sorts of little peaks and things, and they could assume that there were different molecules there. But the amount of molecules that were there, and the fact that they were completely different and not concentrated, and that's very important, is that in order for life to begin, you have to have a concentrated form of something. You can't just have a little bit of glucose over there and then a molecule over there which is something else. They have to be in proximity. They have to be very, very close. Because even if you do believe that if you shake up all these things together, they can eventually form things, they have to be in high concentration and in proximity. They have to be close to each other. They can't be long distances away. And this again is not a really good argument. And I still don't understand why anybody would have believed it. So, and these are the things that we've already discussed as well. The Miller-Urey experiment, of course, it didn't have, as Nick pointed out, didn't have oxygen. Oxygen would have been there anyway in early Earth. Even if this occurred 2,000, 3,000 years ago, the oxygen, as I pointed out, has been going up, not down. I mean down, not up. The oxygen could have come, of course, from photolysis. So if you have water and sunshine, you can make oxygen. And that's fairly well known. And oxygen is a terrible oxidizing agent. That's what the oxidizing agent, it would have messed up all the chemicals that were formed. All the lipids would have been oxidized. And we all know from the news that oxidized lipids are really, really not good for you. That's what causes all the atherosclerosis in your blood. So, and the experiment, as I pointed out, produced the wrong amino acids.
Well, you mean that it produced, you don't mean it produced exclusively right-handed amino acids, do you? It produced a racemic mixture,
Yeah.
half right, half left.
Absolutely, yeah. That's why I said half of what they produced was wrong. So Robert Grange Origins and Destiny in 1986 put out these statements about the experiments. The likelihood of life occurring through a chemical accident is for all intents and purposes 0. So even people that have gone through this and worked out the statistics and worked out the probability is so small that if you put that probability number into, for instance, Bill Gates, everyone thinks Bill Gates is one of the best mathematicians, but I mean, if you put it into Excel, it comes out as 0. So Excel doesn't take that many zeros. It's 0 in Excel. And Miller's beguiling picture of a pond full of dissolved amino acids under reducing atmosphere. This is, this is the ability of just to get enough concentration of the right chemicals in one place is very, very unlikely. And this is from Freeman Dyson, Origins of Life. So, as I pointed out, textbooks still have Miller and Ulrich. I've seen it in textbooks still. It's still there. Suggesting that this is how life began. I mean, we're teaching our children nonsense, not science. Yeah. Even in its day, it was not considered to be an origin of life. It was considered to be a quirky, interesting experiment. There is no mention ever in textbooks of the flaws with the experiment, the fact that it wouldn't work with oxygen present, it didn't have any sulfur around, didn't have any phosphorus around, that the atmosphere on Earth probably wasn't like that. Even the evolutionists don't believe that the atmosphere on Earth was anything like what's in the apparatus there. So I mean, they don't even have people that are supporting this anymore, except it's still in textbooks. So the assumptions are that there were microatmospheres. So what the evolutionists now do is they now try and put the idea that there are very small areas where you could possibly, very minute areas where there could be a concentration of enough of the building blocks to spontaneously combine, which again is now multiplying the absurd by another absurd number. So we already think that there's not enough zeros or not enough time for this to have occurred, even with using the assumptions that the evolutionists use that the Earth is old. There's not enough ATP here. We talked earlier that ATP is what's required for is the energy of life. It's what causes the second law of thermodynamics not to work. It's the energy in ATP that stops things from falling apart in cells, in every cell. And they also assume that the correct macromolecules, all these molecules, somehow coalesce to produce functional units. We know that in the cell, in order for molecules to come together, to be joined together, polysaccharides in plants, or the starches of plants, and proteins, you can keep amino acids in solution for a long, long period of time, and you're not going to have them combine. You need some mechanism for them to combine. The mechanism that's used in cells, all cells, to combine amino acids to make proteins is another protein. So in other words, to get a protein, you need a protein epsilon. And the protein's got to form. So now, again, you have a circular argument. It's not a logical argument to say that an enzyme comes together before it can make a protein that isn't an enzyme. In other words, enzymes are ubiquitous. Every cell has enzymes. Even the virals have enzymes. They have to have enzymes in order to enter the cell. So the assumptions are a little bit on the absurd side. And this is what the experiment shows. It's like having, thousands and thousands of Legos. You go to Legoland and you can have thousands and thousands of Legos. And if you shake them up for long enough, it produces a machine. And the same thing with house. You can buy all the materials you need for a house and you can put them all on a piece of land. And I think that the odds are pretty enormous that it's ever going to build itself without any external force. And again, when the evolutionists say that this does occur, that if you shake the bag long enough it'll form, you can always ask them, well, who shakes the bag? Is it a designer that shakes the bag, or does it just shake? You know, is it and all these things are designed. So what are scientists doing now? So we have problems here. We have a machine that works and uses energy. This is a cell. We need more energy. We need to maintain the machine with new parts, and we need the machine to copy itself. This is what life is. So the cell can be considered a machine that works. It uses up energy. It always needs energy because in order to maintain itself, to stop it from falling to pieces. And it needs new parts to be built, and it needs to copy itself continuously. The other thing that's not thought of with the origins of life is that even if you come up with the odds of one cell forming, in order for the evolutionists to have the LUCA, or the first cell, it has to have very similar cells formed. You never form one. You'd always form more than one. You'd have a lot of these cells together. You can't just have one forming and then suddenly working. You need a lot more, because the odds work in an independent fashion. So if you have enough time, and the time is finite, you need a lot of things will happen the same way in the same time. It's the law of probability, because they're independent events. Then the formation of one would not interfere with the formation of a second.
So, just going on me, just because an organism receives energy, if it doesn't go through the proper channel of the designer's design,